Hello, my name is Tori Hale, and I just want to take a few moments to compare and contrast glycolysis and gluconeogenesis. Now, the objectives of this lesson will be to have the student have a clear understanding of the glycolysis pathway. Additionally, students will have a clear understanding of the gluconeogenesis pathway. Students will be able to identify distinctions between glycolysis and gluconeogenesis. And then students will understand the importance of both the glycolysis and gluconeogenesis pathway. So hopefully by the end of this lesson, you'll have a better understanding not only of the big ideas, but the key things that differentiate both pathways. So to start off, I always like to think, so what? Who cares? Where does this fall in line with things? Well, if you think of cellular respiration, there's three main steps. First, you have glycolysis. Then after glycolysis, you have the Krebs cycle. And then after the Krebs cycle, you have the electron transport chain. So we're only looking at the first bit, glycolysis. And then secondly, gluconeogenesis. So glycolysis. Well, if you literally take the name glycolysis, it's literally the breaking down of glucose. Who cares about glucose? Well, that's our energy. That's how we get our energy, how our cells get our energy so that we can do what we do. So glucose is very important. It's vital for us as human beings to be able to break it down or to raise it up when we need to. In fact, God has designed our bodies very uniquely where our glucose, our blood glucose levels, stay within a very good range. And in part it has to do with the differentiation and the regulation of both the glycolysis and gluconeogenesis pathway. So first, let's just talk about glucose, all right? Because glucose is our ultimate goal. Like, what exactly is it? Well, as I mentioned earlier, it is a chief source of energy. It is found in the blood. So once it gets in the blood, it can be distributed to our cells and be used from there. It's made from food proteins, fats, and carbohydrates. And we'll study it more when we talk about insulin and glucagon. But insulin is necessary for the body to use, and glucose is essential with it. So insulin and glucose kind of go hand in hand, and we'll see that in a bit. Now, if you look at our piece of our slide here, you'll notice that glucose is very similar to fructose and galactose. In fact, they're structural isomers and stereoisomers. So again, from the highlighted regions, we see this double bond between the carbon and the oxygen up here on that first carbon, and then we also see a hydroxide group right there on the fourth carbon as well. So again, it has a unique structure, and there's a specific reason why glucose needs to be this way. Now, if you think about it and we count these out, we got 1, 2, 3, 4, 5, 6. All right, 6 carbons in a glucose. And ultimately, in the realm of glycolysis, we're going to break that down into 3 carbons, all right, that will eventually become what we know as pyruvate or pyruvic acid. So just to get an overview of both pathways before we dive into the minute details, is that glycolysis is a series of reactions. All right, you extract energy from glucose by splitting it into 2 3-carbon molecules called pyruvates. Again, if we just break down the word lysis, means to break. And what are we breaking? We're breaking glucose. Also, it serves as the first step in cellular respiration, as I did mention earlier. And then since the process does not require oxygen, I can have both aerobic and anaerobic organisms use the pathway. So it's very special and I think unique about it that it can in fact use this glycolysis pathway, whether it's anaerobic or aerobic. So oxygen has no dependence on that, and we'll see that when we get into the steps of each one. On the other hand, gluconeogenesis, again, genesis, you think of the beginning, new, all right, glucose. It's literally the making of glucose. As stated in Libre text, it's a metabolic process by which organisms produce sugar. So produce sugar, again, namely glucose, gluconeogenesis, for catabolic reactions from non-carbohydrate precursors. And again, we'll touch on that in just a few moments. Now, a lot of people refer to it as reverse glycolysis. Reverse glycolysis. if we think about that, that's not a terrible way to word it as a reverse of glycolysis, but we'll also realize that there's 3 unique steps that really differentiate it from this glucose power port. So it's not exactly an exact reverse, but you can generalize it that way if that just helps you get the big idea across. So let's continue then and talk specifically about the glycolysis pathway. Again, I know I'm showing you a lot of details. They really break it up into 10 steps, but the key thing you need to realize is that there's an investment phase, and a payoff phase. So you gotta think when you invest in something, you put something forward. The reason why you invest in it is because you hope to get more than you invested, right? You don't want to lose that investment. Same thing with glycolysis. I am going to use 2 ATPs. Why? So I can get 4 ATPs. And then on top of that, I can get some NADHs, which are later going to be used in the electron transport chain. So as we take a glimpse of this, it's really broken down into these 10 steps. But just apart from just mentioning these very complex terms, let's really get into the pictorial representation that I'll zoom up right here. So you'll see right off the get-go, there's 2 things. Again, it's an investment phase. You got to pay off with ATPs because you need ATP to do what you need to do. So we see ATP here and we see ATP here. Again, you'll notice the negative G value, which again is Gibbs free energy, and it makes sense in regards to me having to use this ATP. So as we get to that, and I'll mention more about the negative delta G in just a second, this reaction is going to occur spontaneously into this direction, and it's going to go from glucose to G6P to fructose 6-phosphate to fructose 1,6-bisphosphate, and then you'll notice that it divides into 2 areas. Okay, so it literally splits, and this is where we're really going to start to get a payoff from the investment we did when we put down the 2 ATPs. So again, I said the key idea of glycolysis is you take the 6-carbon glucose and you literally break that 6 carbon down into 3. So I have a pyruvate here and a pyruvate here. Now, as you see both go through this phase, again, as it goes right along here, and you'll notice, boom, an NADH. Again, that's going to be important after the Krebs cycle when you get into the electron transport chain, which remember is the 3rd step in cellular respiration. But then through that, not only do you get an NADH, again, from each of the pyruvates that you're ultimately producing, but you ultimately get an ATP. So this is when we start to get our investment. So we get an ATP back. Again, it goes at this point from 3-phosphoglycerate to 2-phosphoglycerate, all the way to phosphoenolpyruvate, which is PEP, which may be a more popular term that you're familiar with, ultimately to get to pyruvate. And right before you get to pyruvate, what do you see? Another ATP. Now, you might be thinking, according to this graph, I only make 2 ATP. But if you remember, the key is here is that you have 2, right? 2 that are being produced, these G3Ps that ultimately will form 2 different pyruvate molecules. So again, you're making bank. You're making a net total of 2 ATPs. You're getting NADHs, which are later going to be used in the electron transport chain so that you can get even more ATP. And then after that, you're good. Again, the first step of cellular respiration, the breakdown of glucose. So again, glycolysis, if you look at the nitty-gritty details, it may seem super complicated, but if you try to get the big idea first, the names kind of fall in place. So that's an overview of the glycolysis pathway. Again, I'm using only 1 glucose for 2 pyruvic acid molecules, or pyruvate. Even though you have to initially invest 2 ATP, you end up getting 4 ATP, which again, you get a net of 2 ATP. And then you get 2 NADHs, which are going to be used later in the electron transport chain, and you're going to get even more payback, even more ATP. Then ultimately, we're going to talk about, as we'll relate this to insulin, how the insulin, all right, actually promotes glycolysis in our body. So insulin promotes this breaking down of glucose. Conversely, let's talk about the gluconeogenesis pathway. Again, I'm not breaking down glucose. I want to make glucose. Gluconeogenesis. I want to make it. So again, don't let the picture fool you or overcomplicate things. But ultimately, as you can see right here, we have our glucose. Again, glucose to G6P, G6P to fructose 6-phosphate, fructose 1,6-biphosphate. Oh, if I go in this direction, what does it look like? It looks awfully like glycolysis. But what separates gluconeogenesis? It's because, and if you notice about these arrows, it goes this way. All right, so this is why people say it's the reverse mechanism. Now, I'll point off a few key things that clearly differentiate it, so it's not a clear, distinct reversal, but you'll see it's very, very similar. So, as we go through, we have our pyruvate. Again, does that make sense that we started off with our pyruvate? Yeah, because remember, at the end of glycolysis, we ended up having 3 carbons, which were pyruvate or pyruvic acid. From there, it gets into the PEP that I mentioned earlier, 2-phosphoglycerate, 3-phosphoglycerate, 1,3-bisphosphoglycerate, glyceraldehyde, fructose 1,6-biphosphate, fructose 6-phosphate, glucose 6-phosphate, and glucose. All right, again, very, very similar to what we saw before. But a couple things. And I'll scroll back up at our former drawing of glycolysis just to point things out. So if we're looking at this, and I'll erase all my drawing to get the key things apart, there's a couple areas that are going to be trouble for us. So let me highlight them. First of all, we think of this negative delta G value, this negative delta G value. All right, and we have these endings of these negative delta G values. What's the thing with negative delta G? That's a spontaneous reaction. If you do the reverse, that makes it a positive delta G, making it a non-spontaneous reaction, which means it's not chemically favorable to occur. So we're adding a little bit of our chemistry knowledge into this. So you have to find a way around it. You have to find a way to get through those points that are non-spontaneous to ultimately get what you wanted at the beginning, which was glucose. That's why, near the end here, this is why they're going to use OAA, all right? Because through that, they can get to their desired phosphoglyceraminase, all right, and so on and so forth, to produce the ultimate result of glucose. Again, in gluconeogenesis, you're going to have to overcome these 3 humps, all right, that initially aren't so chemically favorable, but you're able to use enzymes to overcome it. So, as we look at this, I want to get some key takeaways. All right, number 1, gluconeogenesis is virtually the reverse of glycolysis, except for 3 steps. And again, we'll see this more detail on the next slide. We know that low blood glucose will stimulate this pathway. Why? Because I have low glucose, so my body needs to actually produce glucose. All right, the hydrolytic reactions, and again, we'll see this more on the next slide, make this process unique. Interestingly, in animals specifically, glucose cannot be generated from acetyl-CoA and fatty acids. And I also want to mention, as stated by Gonzaga, that substrate cycles opposing reactions allow large change in net metabolic flux and are thermogenic. And then ultimately, glucagon promotes gluconeogenesis. So I mentioned earlier about how our body and God has created us in such a way to have a balance between insulin and glucagon. And if insulin promotes glycolysis and glucagon promotes gluconeogenesis, they're going to work hand in hand to keep our body in balance and in equilibrium so that we can maintain a very good level of blood glucose. So, before I go more into the application of insulin and glucagon, I do want to just drive home and do a side-by-side comparison. So, let's look at a side-by-side comparison of both glycolysis and gluconeogenesis. So glycolysis is on my left in an orangish reddish tint, with gluconeogenesis on my right in that bluish tint. So you'll see right off the get-go, as mentioned, we have our ATP, which produces ADP after it's done. Again, if we're sticking with glycolysis, we have another ATP because we're going through the investment phase, if you remember. And this will allow us to generate our NADHs, our 2 ATPs, technically twice, since we have 2 molecules, since we broke down that 6-carbon glucose into a 3-carbon chain. So we get our 2 ATPs, and bippity-boppity boom, we get our 2 pyruvates. All right, again, we say that it's virtually the reverse in the sense, and I'll highlight this in a different color. We start off with our 2 pyruvate, right? And we're going the opposite way. But I just can't go straight to here. All right, so in fact, if we think about this, it uses 2 ATP and 2 GTP through what? What I mentioned earlier, OAA. I told you we were going to get back to it. So oxaloacetate. So it's going to go through this oxaloacetate to get back to my PEP. So that's one hump, and remember we said there was 3 that we had to override. Why? Because it ultimately had a negative delta G. When I reverse the reaction to get from pyruvate to PEP, that's a positive. So I have to find a way to overcome that unfavorable situation. So again, very similar. As you go through, you see a lot of these arrows able to go both ways. That's because the delta G is so low, it's virtually 0, so they can easily cross between both reactions. And then ultimately you have to come through a couple of those humps. So again, we have our 2 ATP here being turned into ADP, basically mirroring the other side. We have our 2 NADHs now becoming 2 NADs. And then here we go, we have our water undergoing, and we're producing an inorganic phosphate. And then same thing on the other side. And again, why do we have to do that? Well, we spent 2 ATPs here. And again, you got to think about it. Those 3 main areas where I had a negative delta G, when I do the reverse and use gluconeogenesis, I have to find a way to make it chemically favorable. So you're going to have to find things, like in this case, the glucose 6-phosphatate, or the fructose 1,6-biphosphatase, or bisphosphatase, to overcome that and to kind of go around that unfortunate situation. But ultimately, this is how you put the 2 together, all right? They go right in sync, hand in hand, virtually the reverse of each other. Now, application. So you might say, great, I see this complexity in my body, so what? Well, the great thing, and just maybe some interesting facts for you to see the bigger picture, is that glucose provides 3.8 calories per gram. 3.8 calories per gram. Now, what's interesting is that we do not have to directly consume glucose. If we wanted to, our body, as long as we have the proper ingredients or the proper precursors, we can create glucose. And you know that just simply by understanding gluconeogenesis. That, in fact, shows that we don't need direct intake from glucose. We can just have the ingredients to make it and then make it ourselves in our body. And again, some of those ingredients would be stuff like proteins, let me highlight them here, proteins, fats, fructose, or even galactose. And when you think about that, let's see here. In fact, I still have some Halloween candy in here. We even have dextrose right here in my little gummy wrapper. So there's tons of ways of which you can actually make your glucose, even if you don't directly take it in. Now, there are some ways that you can get glucose directly in. You can see it in the picture, specifically fruit, honey, sweetener, syrup, things like that. That would be a great way to get a direct intake of glucose. But again, it's not necessary because our body can always make it from the ingredients. So, I mentioned earlier about this way that we have this regulation between glycolysis and gluconeogenesis, and I kind of hinted at this idea of insulin promoting glycolysis and glucagon promoting gluconeogenesis. And this is hand in hand. I like to think of it kind of as following a macro level of Le Chatelier's principle. And if you remember Le Chatelier's principle, it's basically that ultimately these reactions, your system wants to maintain equilibrium. So if it ends up shifting more to the products, it wants to create more reactants, depending on the pressure, on the temperature, you name it. There's various factors that are going to affect the tipping of the scale, so to speak, but ultimately you want to maintain equilibrium, so your body's going to adjust and the system's going to adjust so that it can try to remain a good balance. Very similar to this idea of insulin and glucagon and gluconeogenesis and glycolysis. Again, it follows this Le Chatelier on a macro level to try to balance things. Do I need more glucose? Do I not? And it also kind of helps explain why we're able to maintain such a good blood glucose level. So without this type of regulation system, we wouldn't be able to maintain the blood glucose levels that we do. So, just some highlights about insulin. It promotes glycolysis, as I mentioned, and it's an anabolic hormone, anabolic. So it builds up. And ultimately, if I have insulin in my body, since it promotes glycolysis, it's going to break down glucose. And that's why it's going to decrease glucose levels. So if I want to decrease glucose levels, I want insulin. I want insulin to encourage and promote this idea of glycolysis. Conversely, if I'm looking at glucagon that's promoting gluconeogenesis, it's catabolic. So again, it's the opposite of anabolic. It doesn't really build up, but in fact it breaks down. And what is it breaking down? Well, it breaks down glycogen and ultimately increases glucose. And does that make sense? Yes, because glucagon promotes gluconeogenesis, which is creating more glucose, which means there's going to be an increased level of glucose in our body. So these are two instrumental pathways that help maintain our blood sugar level. So very, very, very important. So hopefully from this video, as you're looking back between these two pathways, hopefully you have a better understanding about the glycolysis pathway. Again, a simple breakdown. I take a 6-carbon glucose and I break it down into a 3-carbon pyruvate and another 3-carbon pyruvate. Again, I put an investment phase of 2 ATP, so I have an overall 4 ATP, 2 NADH just to be used later on in the electron transport chain, and then ultimately the 2 pyruvates. Secondly, hopefully you have a better understanding of gluconeogenesis, which again is just the making of glucose, virtually the reverse of glycolysis. However, through these objectives, hopefully you notice the distinctions. Those 3 main areas that are not chemically favorable simply because of the Gibbs free energy value. Again, the other ones are really of no concern because the delta G is basically negligible, so it can easily reverse between the substances. But fourth, through it all, hopefully you understand the importance. So same thing by diving into insulin and glucagon, hopefully you're realizing that it's more than just something in a textbook, but something that applies to real world. And in fact, when you study even more, you can figure out the diseases or what could go wrong in the body by figuring out if there's a mishap throughout one of the processes. You know, what if you don't produce that pyruvate? What if you have a mishap or you don't have enough ATP? There's different diseases associated with different disorders in those particular pathways. If you want any more information, again, feel free to reference this slide, which contains all the places that I got my info on. But most importantly, I just want to make sure you get the big picture. If you don't remember anything, glycolysis, literally by the name, the breaking down of glucose. However, gluconeogenesis is the synthesizing, the creation of glucose. And that's why our bodies, we don't have to directly ingest glucose, because we can have it and we can create it in our bodies if we have the necessary precursors. So hopefully through this you have a better understanding of glycolysis and gluconeogenesis. Hopefully you realize the importance of it, that it goes beyond a textbook or just a simple assignment, but that it really has real life and real world applications. Because again, not to lose sight of the big picture, glycolysis is one step out of our cellular respiration. Remember, after glycolysis, it gets fed into the Krebs cycle, which OAA is a part of that, and then after that, the electron transport chain. So even though I invested a small amount, I gained 2 ATPs, but guess what? Even later on, you're gonna gain more through this breaking down of glucose. And it's why it's so important that we have glucose, that we have energy for our cells. Otherwise, we wouldn't be able to do the things that we do. So hopefully that gives you a better knowledge. I hope that the references are also an excellent source. And maybe through it all, you appreciate the glycolysis and gluconeogenesis pathway just a little bit more.